Yeah.
Okay, so today we're going to continue with our understanding and study of the gastrointestinal system. When we left off last time, we had talked about how we break down, digest, and absorb our carbohydrates starting in the mouth. We talked about protein breakdown and digestion. And then we moved on and talked a little bit about lipid digestion and absorption and how that's different because of the different chemical properties of our lipids. So we were thinking about enzymes, where they're secreted, and what they can do. Now, to finish up this topic of absorption and how we're getting nutrients and essential components for making things in our cells out of our food, we're going to think about vitamins and minerals. Okay. So we're not going to have to worry about breaking them down. We're just going to be thinking about how we physically pull them into the body, how we absorb them. So depending on what vitamin we're thinking about, we're going to have different mechanisms for that absorption. So when you're talking about vitamins, we actually separate them into fat-soluble vitamins and water-soluble vitamins. So that's going to depend on their chemical properties, what they mix best with, right? So we know our fats, our lipids, are non-polar, they're hydrophobic. So some of our vitamins interact well with those, and our water-soluble vitamins are able to dissolve in water. They interact with that polar environment from our water molecules, hydrophilic. So they're going to have different transport mechanisms, essentially. Most of the vitamins we think about are fat-soluble vitamins. So vitamins A, D, E, and K are fat-soluble, and they're just going to get absorbed with lipids. So when we were talking about lipids, we saw that we had these big fat globules that were broken down into smaller fat droplets using our bile salts, and then they're forming some micelles, but also having components of those droplets cross across the membrane, just directly dissolve through after they're broken up by the lipases coming from the pancreas. So all we want to know about fat-soluble vitamins is they're just kind of pulled along with that process. So they're moving along the lipids. So you'd expect to see these fat-soluble vitamins in the lipid droplets themselves. You'd see them mixed in with the micelles hanging out in the lumen, waiting to be absorbed. And then you'd see them in the chylomicrons as well. Those were the things that we were creating inside the cells in the walls of the GI tract that are eventually getting moved into our lacteals and into our lymphatic system. So that's what we were ultimately making with the smooth ER and the Golgi moving into the body. Okay, so that's our fat-soluble vitamins. For our water-soluble vitamins, you're going to have specific transport proteins for those specific vitamins. So imagine just like when we had channels for sodium, channels for potassium, transporters for glucose, you'd have a special transport protein dedicated to each water-soluble vitamin. One that we pay specific attention to, or a vitamin we pay specific attention to, is vitamin B12. Vitamin B12 is necessary for synthesizing hemoglobin. So if you don't absorb enough vitamin B12, this can cause anemia. One of our types of anemia was pernicious anemia. That's caused by a deficiency in vitamin B12. And the thing about vitamin B12, we are going to have to transport it through, but it's also going to need a second sort of helper molecule. So vitamin B12 only gets absorbed if it's bound on to something called intrinsic factor. So really, number of ways you could have vitamin B12 deficiency for you, yourself, your body, right? One, you're not eating enough vitamin B12. Two, you're not producing enough intrinsic factor, things like that. So a number of things could go wrong. So for minerals, we're thinking really ions. So sodium, chloride, and potassium. We want to absorb these. We know that we need these ions, these minerals, in order to drive a lot of processes in our cells. So for sodium and for chloride, I guess, since they go together, a lot of the sodium absorption is going to happen as something we call solvent drag. So as we reabsorb water, sodium is going to kind of follow along with it. So this is more going to happen earlier on in your digestive tract. So think like in your small intestine. Most of our sodium absorption that we're focusing on, active absorption, is going to happen in the jejunum and the ileum. And then some happens in the colon, but that's more of a process similar to what happened in our kidneys, where we're thinking gradients and stuff. Okay. Chloride has an opposite charge, right? Sodium has that positive charge. Chloride ions have a negative charge. So chloride follows that sodium because of that attraction of that electricity. Potassium gets passively absorbed. An interesting thing about potassium is that what exactly happens with potassium is going to depend on the concentration in the lumen of the GI tract. So that chyme, all that mushed up food and juices moving through your small intestine and then your colon. So if you have enough potassium, you're going to absorb it. But when the concentrations of potassium in the lumen and that fluid or semi-solid moving through are really low, potassium actually gets secreted. So it moves out of the cells lining your GI tract and into the lumen. So this means that in situations like diarrhea, where you have watery fluid moving through your GI tract, low concentrations of things like potassium dissolved in it, you can end up with potassium secretion, removing potassium from the body, and therefore creating low potassium hypokalemia in the body in the plasma. Continuing, one more thing that we want to pay attention to is bicarbonate. So we know that bicarbonate, we need it in our plasma to act as a buffer for our pH, which is important. We want it to be able to kind of soak up any extra acidity, either coming from that carbon dioxide or coming from excess hydrogen ions. So we are going to absorb bicarbonate moving through our GI tract. So as we're moving through, so when you eat something with a bunch of baking soda in it, for example, that's a source of bicarbonate. Go through your mouth, go through your esophagus, your stomach, in your jejunum, it's going to start to be passively absorbed. When we're in our ileum and our colon, we are going to be exchanging some of those bicarbonate ions for chloride ions. So that's an additional way that we get chloride into our body, in addition to moving it along with the sodium. For calcium, calcium is going to get actively absorbed in the duodenum and the jejunum. And the way it does that is it binds to a protein embedded in the brush border. So the brush border was that series of microvilli on the apical side of those enterocytes, that lining of the GI tract. It's all furry because of those projections. So we have proteins for the absorption of calcium there, called calcium binding protein. This is going to transport our calcium into our epithelial cells, that lining of the GI tract. And fun fact, calcium absorption, vitamin D helps with that. I think you've probably heard that out in the world. If not, you'll notice if you ever take calcium supplements, they're usually combined with vitamin D. That's because it basically helps calcium binding protein, helps you make more of those, things like that. So that's why that improves our absorption of calcium. So once we have our calcium going through that calcium binding protein into the lining of our GI tract, we can transport it across the basolateral membrane using a calcium pump. So we'd use ATP, we'd use energy to do that. Now we have iron. Iron was another important component of hemoglobin. We know that's in the center of each of the 4 heme groups, allowing us to bind on oxygen, carry oxygen through our cardiovascular system. So the way we're going to move the iron around is with something called transferrin. So when we saw this, and when we were talking about hemoglobin before, we saw T iron and F iron in an image. So T iron was the transferrin with iron. F iron is a storage molecule, so that's ferritin. See that there? So just linking those 2 terms up. Okay, so the way we can move iron around, absorb it into our body, is that some enterocytes, so some cells lining our GI tract, are going to secrete transferrin into the small intestine, which can then bind the iron, so forming that T iron. So that's representing a transferrin iron complex. So they're joined together. Okay. Your cells recognize this complex, this transferrin iron complex. And so when your cells come into contact with that T iron, those 2 things bound together, there's a receptor, notices it, and then you're going to move that whole complex into the cell through endocytosis. So we have essentially our little cell, we'd have a receptor, it would notice the T iron. Okay, all right, so those are bind in, and then we pull it in by kind of like bubbling off this membrane, breaking off the bubble inside so that you have the T iron in the cell. Okay. So from there, you have a couple options. So some of that iron is going to get stored as ferritin, so that iron, so it can just hang out until you need it. Some of the iron is going to move directly into the blood. So this process would happen just basically on the other side of the as well. So it would stay as that T iron complex, that transferrin iron, but then it would move into your bloodstream, into your plasma, get around to where it needs to go. For example, to your bone marrow to produce more red blood cells, give them their hemoglobin. For water, there is a lot of water in your GI tract. Honestly, even if you are not drinking as much water as you maybe should be. So typical intake for water should be about 2 liters per day. But there's a lot more water because of all the secretions into the GI tract. So as we're secreting enzymes, things like that, into the GI tract to help digest food, that is going to involve water for those enzymes to be mixed around in. So all those secretions add up to about 7 liters per day. So you're going to want to reabsorb a bunch of that water. This process is passive, and it's going to essentially look pretty much like it did when we were thinking about the kidneys. It's going to follow along some solutes by osmosis. So you'd expect to see osmotic gradients, water moving along as we shift those gradients. We're not going to talk in detail about setting up gradients like we did in the kidney, but it's an analogous process essentially. These are questions for you to think about for later. We're going to move into control of the GI system. So when we think about controlling the GI system, what we're really thinking about is like, how do you get from smelling a tasty burger and eating it to actually producing all those enzymes, digesting it, absorbing it? How does your body know to do those things? We also might want to think about, where do hunger signals come from? Or like, why do you feel full? Right? So this is going to be combining a bunch of different parts of your body. So we're going to have your nervous system communicating between your mind, so in your head, giving you signals in the stomach, We're also going to have signals coming from the small intestine. So we're going to be getting feedback, both from what we see, smell, our senses, as well as physical feelings as food moves through our system. And we're going to have various reflex loops running around that control what enzymes you're releasing, essentially through signaling hormones and some local sort of paracrine factors. Yeah. So when we think about phases of gastrointestinal control, it annoys me a little that they use the word for phases, because I'm not sure that that's super obvious, but essentially what we're thinking about, like, think about smelling some great food that you really like. And we're going to think about like a timeline, a process of what happens as you go eat that food. So the first thing that's happening is you're sensing that food. So you're going to think about the food, you're going to smell the food, you're going to taste that food. All of that is sensory information from your head. Anytime you see this root, Seth, that refers to the head. I think it must be Greek. So the cephalic phase of gastrointestinal control all has to do with stimuli originating in the head, aka far away from your gut. So when we are thinking about things that are under cephalic phase control, we're going to be thinking about long reflexes, because we're going to have signals traveling from your head to your GI tract, to your GI system. So we'll talk about what long reflexes and short reflexes are in a bit. But we're going to have to travel a long way if we're getting from our head, so we can know right off the bat that these are going to be long reflexes. They're going to need input from the central nervous system, which, if we think about it, right? How do we taste things? How do we smell things? That comes back to our cranial nerves going into our brain, right? So we need input from the central nervous system because we have that relationship with the cranial nerves as our sensory nerves coming into centers for our brain for processing that information, and then finally sending out signals to the rest of the body. So long process, long reflexes. Our other two pieces of gastrointestinal control are going to be a bit more local. So once you taste that food and swallow that food, you're done with this cephalic phase control. But we are going to be digesting that food. That food is moving down your esophagus, moving to your stomach. The word gastric always refers to the stomach. So our gastric phase control is all going to be about stuff coming from the stomach. So once the food reaches your stomach, we're going to have stimuli that the stomach is releasing to say, hey, we've got food in here. Actually, we can also sense when there's no food in the stomach. Okay. So this is going to be involved in both long reflexes and short reflexes. And your stomach is also going to be releasing gastrointestinal hormones. So it's going to be releasing signals into the blood about this. We're not done with our digestive process once we hit the stomach. We are then also moving that food into the small intestine. So the duodenum, the jejunum, the ileum, and then into the colon. Okay. So we are also going to have intestinal phase stimuli. And so intestinal phase control. So we're going to have that involved in, again, both long and short reflexes. And we are also going to have GI hormones released in response here as well. So the stimuli in the intestinal phase are going to be coming from the small intestine. So that first part of our intestines is where we're going to start to see that signaling. Yeah. So all we need to know about these phases is where is the stimulus, and that tells us what phase we're thinking about. So we're going to walk through thinking about what we're secreting and how we control that. So when we're thinking about our saliva, right, it's released by our salivary glands into our mouth. And we know that for our different macromolecules, saliva is one of the places that we're going to begin digesting carbohydrates specifically, right? We saw that salivary amylase was important to beginning off that process of carbohydrate digestion. Okay, so we are going to want to control whether we have enzymes like salivary amylase in our saliva ready to digest carbohydrates, or whether we want to have a more sort of watery saliva, just kind of keeping our mouth moist. Okay, so we are going to actually produce different things in the saliva depending on whether we think we're about to eat something or not. Okay. So when we're thinking about saliva secretion, the sorts of stimuli that we're going to think about are things like the taste of food, the texture of food. We're going to touch receptors in the mouth. So mechanoreceptors, think mechanical, right? That's physical sort of touch. And chemoreceptors, so response to the chemicals. So both of those are going to be able to sense that you are physically chewing food, essentially, that there's food present in your mouth. Sight and smell input would also have an effect on saliva secretion. So our other cephalic phase sort of mechanisms of control. So when you see food, when you smell food, and if you think about food, that's running around in your cerebral cortex. We have that information. There's a salivary control center. So that can also stimulate saliva secretion. Okay? So if you feel your mouth literally start to water, it's doing it now that I'm thinking about food, right? That's from that control from your cerebral cortex. Okay. So when we're thinking about control of what the contents of the saliva are, we're going to be thinking about our autonomic nervous system again. So we're thinking this is an automatic, autonomic kind of response. Okay. So our parasympathetic activation is actually going to produce watery saliva, while our sympathetic activation is going to produce protein-rich, viscous saliva. Okay. So we're eating our food. Now it's getting down to the stomach. Right? And the stomach needs to know whether it is going to produce a bunch of stomach acid, whether it's going to produce pepsinogen so that we can start digesting protein. Okay. So for acid secretion, this is going to be in response to activation of the parasympathetic nervous system at rest and digest. Okay. We also have a hormone gastrin that is going to create acid secretion, as well as histamine, actually. So histamine in the stomach actually acts as a paracrine factor, so like a local signal, whereas in the rest of the body, it's doing things like inflammation, right? It does have a specific local role in the stomach, which is to trigger the release of acid as well as pepsinogen. We can see it's on that second list as well. Yeah. So the pepsinogen secretion is happening at the same time as secretion of the stomach acid, which is handy because that pepsinogen has to be activated by the acid. And pepsinogen is the inactive form. We have to sort of denature it, start to break it down using the acid to turn it into pepsin, which can actually start to digest our protein. Okay. So we're going to have the same triggers for that secretion. We're going to have parasympathetic nervous system, our hormone gastrin, as well as histamine. So thinking a bit deeper about this. So this is actually going to begin with some cephalic phase regulation as well. Okay. So cephalic phase stimuli that are going to increase the secretion of acid and pepsinogen. So the two are generally going to go together. Be things like the sight of food, the taste of food, the smell of food, as well as the feeling of chewing and swallowing, right? So essentially from those mechanoreceptors, maybe the chemoreceptors in your mouth and in your throat when we're thinking about swallowing. All of this is going to activate the parasympathetic nervous system. And then that parasympathetic nervous system has an effect down on the stomach, which is to stimulate the gastrin, which we saw in that previous slide as one of the more immediate ways that we're signaling the release of acid and pepsinogen. Yeah. So we're going to have a flow chart here. So we're looking at cephalic phase control of gastric secretions. So what we have, we're seeing, smelling, tasting food, we're chewing and swallowing. This is upregulating parasympathetic activity, so that rest and digest. We're having an increase in the activity of that part of our autonomic nervous system. Okay. This does have a direct effect on the parietal and chief cells in the stomach, telling them to release more acid and pepsinogen so that we can start digesting food as it comes in. And it also has this sort of secondary little pathway, increasing this response, where it goes to G cells. These are in our stomach as well, which release gastrin, which is a GI hormone. That gastrin goes into the plasma. Okay. And that gastrin traveling around in the plasma is an additional signal to the parietal and chief cells to release acid and pepsinogen. Okay. So this is how we kind of pair together signals coming from the head, so cephalic phase stuff, with some of the more local actions of our stomach, some of the more gastric phase stuff. So when we're thinking about stimuli coming from the stomach itself, to which the stomach can respond, we're going to be thinking about things that would signal the actual presence of food in your stomach. So when we're thinking about gastric phase stimuli, we're going to be thinking about your stomach sensing that there are proteins, peptides, and amino acids in your stomach. So for gastric secretions, we're really interested in protein digestion specifically, because it's that turning pepsinogen into pepsin by activating it using that acid that we're concerned about in the stomach, right? Stomach is really the start of protein digestion specifically. Okay. It's not the place all of our protein digestion happens, but it's the first part, right? Okay. So when we sense in the stomach with other receptors that we would have, that we have these products, the proteins, the peptides, the amino acids, we're going to start to secrete more gastric secretions, so the acid and the pepsinogen. We also have mechanoreceptors, so like physical touch stretch sensors that can sense how large your stomach is getting. So when we start to stretch out the stomach as it's getting full, those rugae, those wrinkles are expanding. Okay? Your stomach can physically sense that it has grown larger, that it is filling up. So that is also going to be a stimulus for producing more of these secretions. Okay. So we have a sort of longer pathway for triggering gastrin, which we looked at in that flow chart, and it had to do with those cephalic phase stimuli. But we also have this shorter sort of reflex pathway where your stomach senses that there's proteins and broken down proteins in the stomach, and then immediately produces more acid and pepsinogen. And so it's kind of like a local response for the short pathway and a longer response going back to your central nervous system for that input from the cephalic phase. So here we see how that works. So we have our proteins and pieces of broken up proteins in the stomach. Okay. And we have that physical sensation of the stomach expanding, that stomach distension, as our initial stimuli here on this gastric phase control. Okay. So you have chemoreceptors detecting those proteins, mechanoreceptors sensing that distension of the stomach. And their response, some of it's going to be real immediate, increasing acid and pepsinogen secretion, right? Some of it's going to take a longer time. So we're going to have some parts going to the G cells in the stomach, producing gastrin, our hormone, going into the bloodstream, becoming plasmagastrin, circulating back to the stomach, and producing more acid and pepsinogen. Now, we don't want to produce these secretions in great quantity all the time, right? That acidity of the stomach, if we secrete too much acid, we're going to make it more and more acidic. That's uncomfortable and could start to damage our stomach itself. And we don't want to be hungry all the time. So we also do inhibit secretion of our gastrin, our acid, our pepsinogen. After the stomach begins to shrink back down, we begin to release our food into the duodenum. Okay. So in the stomach, our food has been mixing around with the acid, with the pepsinogen, which is turned into pepsin. We're breaking down some proteins. We're now calling it chyme because it's a bunch of stuff all mixed together. Okay. That chyme is going to start slipping into the duodenum, going out our pyloric sphincter. Okay. So as that food is exiting, we're removing a bunch of stimuli for the secretion. So particularly, you can think about like distension of the stomach, right? So it was bigger, distended. Now that we're moving stuff into the duodenum, it's shrinking back down, it's getting smaller. So we've removed that stimulus that was telling us to secrete more acid and pepsinogen. Okay. And also, now that we're not eating any more food, right? At this point, our meal is over, right? So the acid that we are producing, right now that it doesn't have food to mix with anymore, is increasing the acidity of that environment in your stomach. So that increasing acidity itself is actually going to be inhibiting the release of gastrin, that GI hormone. That was one of the ways we were kind of increasing our response to the food. Now, it's also the case that our food, as it's leaving the stomach, right, is moving into your small intestine, into the duodenum. Okay? So we are also going to be able to sense, able to notice that food moving into the duodenum. So as that food moves into the duodenum, it's going to be increasing the osmolarity of the contents of the duodenum, right? So we've got more stuff in it, not just water. It's going to be increasing the amount of fats, the amount of acid in the duodenum. So any fat you ate and the acid coming out of the stomach mixed in the chyme, right? That's now in the duodenum as we're moving through our GI system. And the duodenum itself is filling a bit, right? So we can also have some distension of the duodenum. So some stretch, some expansion there of that part of the pathway. Okay. So these signals coming from your intestine are going to sort of loop back and inhibit secretion of these products from the stomach as well. So we're noticing that the stomach is emptying because we're not stretched out anymore. It's getting more acidic in the stomach because we're having acid buildup. So that's a signal that maybe we don't need to produce any more acid and pepsinogen. And at the same time, because the food has moved as chyme mixed with those gastric juices into the duodenum, we're sensing the presence of food, fats, of these broken down bits of protein in the duodenum, maybe the duodenum stretching a little. So that's another signal that the food has moved out of the stomach, essentially. So we're going to have pathways looping back, inhibiting acid and pepsinogen secretion from the stomach. So we're going to move on to thinking about why we are secreting pancreatic juice and bile. Okay. So when we think about the shape of our gland, we want to think about, and we can draw them a little bit like alveoli. They're not filled with gas, though. These are just our cells. So these are acinar cells, acini. Okay. So we're going to have some of those cells. And then we're going to have a duct where we have stuff travel. And there are also duct cells. Okay. So that's the general sort of breakdown of what you might see if you took a slice of a gland and sectioned it and looked at the histology. Okay, so we got our acinar cells and we've got our duct cells. They're going to be secreting different things. So the acinar cells are going to be secreting a small amount of what we call primary secretion. So we'll take a look at what that is in a second. They're also going to be secreting water, electrolytes, and digestive enzymes there. And then in the duct cells, we're going to have a large volume of pancreatic juice stuff, and it's going to be bicarbonate rich. Okay. So the main things we want to think about when we're thinking about the control here of pancreatic juice secretion, the bile, is something called CCK and something called secretin. Okay, that one, you know, it makes you secrete stuff. They called it secretin. I like that naming convention. That's very good. CCK, not so much. All right. So whether we are secreting enzymes and or bicarbonate, it's under a little bit of different control. So you could, in theory, secrete a bunch of enzymes, but not much bicarbonate, or vice versa, because we have slightly different signals to these different cells. Okay. So we have independent regulation of these two main parts of our pancreatic juice. We know we need those enzymes to help us continue to break down proteins, right? We had a bunch of zymogens we were secreting that were moving into the duodenum. We were activating them to continue to break down proteins. And we need that bicarbonate because the chyme moving into the duodenum is acidic because of all that stomach acid. We want to protect our small intestine, so we want to kind of neutralize some of that with our bicarbonate stuff. So the composition of the juices coming out of the pancreas, the pancreatic juice, is going to vary a bit based on the contents of the lumen. We also had, for example, our lipases were coming from the pancreas. So they were helping us break down fats, right? So we might want to know whether we want to break down a bunch of protein, whether we want to break down a bunch of fat, what exactly kind of proportion we would want there. So the stimulus for secreting enzymes is mainly going to be the CCK. Okay. So it stimulates the acinar cells, so those little globes kind of at the end, to secrete our various enzymes for the breakdown of proteins and fat. Okay. And it's a weak, essentially, stimulant for the release of bicarbonate. So it doesn't not signal that you should release bicarbonate. It's just not very good at it. So we really want to just like associate it mainly with enzymes. Meanwhile, the secretin is our main signal for secreting bicarbonate. Okay. So that is happening mostly from our duct cells. Okay. So cells of that kind of tube, that little pathway on the way to our duodenum. Okay, so secretin, bunch of secretin, you're going to be secreting a bunch of bicarbonate into the duodenum. Now, you're going to notice that on this slide, we have this word potentiation. Okay, so we do want to associate CCK with enzymes, secretin with bicarbonate, but each of them is a weak, a sort of small stimulus for the opposite function. Okay. And the presence of one helps us kind of do the opposite as well. So CCK and secretin have effects on each other. And that's really all that means. So we're kind of prepping the opposite one as well. Okay. So primarily, CCK is helping us secrete enzymes, but it's also helping us kind of activate and strengthen this other response, secretin telling us to secrete bicarbonate and vice versa. So we're regulating enzymes and bicarbonate separately because we have these two different enzymes with two different main roles, but they do kind of play off each other as well. So we have different stimuli for the release of each of them. Okay. And these are really just related to what the job of each of these secretions is. So CCK is telling us to make more enzymes in the pancreas. So the signal for that is going to be, we have fat and amino acids in the duodenum that we need enzymes to break down, right? So CCK helps us release enzymes. If we sense that we have fat amino acids, we know we want to break them down. We know we need those enzymes, right? So that's our stimulus for the release of CCK versus our stimulus for the release of secretin. Secretin tells us to secrete bicarbonate, right? So the stimulus for that is that our duodenum is getting pretty acidic and we want to essentially buffer that out. So we're going to secrete secretin, which is then going to help us secrete bicarbonate. The reason these two things might go together at the same time is as we start to pass that chyme through, it has both stomach acid in it as well as whatever food you've eaten. Okay. So we have sort of feedback loops here that we could look at. So if you see increasing acid in the duodenum, you'd have chemoreceptors that would detect and respond to that. There would be some nervous pathways, both sort of local and longer, going up to your central nervous system, which would then go to our endocrine cells, so cells that produce hormones, telling us to release secretin. The secretin and acid go together. That secretin would get into the plasma, get to the duct cells of the pancreas, telling us to secrete bicarbonate in order to buffer out that acid, essentially. So we neutralize our acid in the duodenum once we have that pancreatic juice there, and the acidity of the duodenum drops. So this is a negative feedback loop. Once we've neutralized it, this whole signal is essentially turning off. So pancreatic juice, great. You like that it has enzymes. You like that it can break down proteins, can break down our fats. It also has its own amylase in there. So it does continue kind of finish up the starch digestion that began in the mouth with your saliva. Okay. But in order to really digest all those fats, we also need bile, right? Because we need those bile salts to break up our big fat globule into smaller little fat droplets. Okay. So the regulation for that secretion of bile, which is coming from our liver or from the gallbladder where it's been stored after being made by the liver, we're going to have both secretin and CCK involved in that. Okay. So secretin is going to stimulate bile secretion from the liver directly. Okay. So it's just telling the liver, okay, give me your bile. That's the secretin. That was the one that was helping us release bicarbonate from those duct cells of the pancreas. CCK is going to run up to the gallbladder and tell the gallbladder that it should contract. So when the gallbladder crunches down, right, squeezes, anything that's in the gallbladder gets released. Right, so as we release the contents of the gallbladder, that bile then moves through our cystic duct, links up into our common bile duct, and dumps us into the duodenum as it goes out the sphincter of Oddi. So CCK is also stimulating the relaxation of that sphincter, so like opening up that duct essentially, so that we can get this bile into the duodenum. So these two things, again, are working together to get bile into the duodenum so that we can break up those fats. We can start to digest them with our lipases coming from our pancreatic juice. Okay. This is also partially why you don't necessarily need a gallbladder, right? Like it's helpful. We do need those bile salts to kind of break up those big lipid droplets, but we do also get bile directly from the liver. We can see that being stimulated by the secretin. So if they remove your gallbladder, it's not like you have zero bile salts available to you. So here we see just a pathway map of some of these things. So we see the signals in the duodenum that we saw before. So it's getting more acidic because we have that chyme, which is stomach acid mixed with food, coming into our duodenum. We have broken up proteins. We have fat droplets coming into the duodenum as well. So those actual molecules. The acidity is telling us to release secretin, a GI hormone. The proteins and fats are telling us to release CCK. These have primary roles. Secretin helps us release bicarbonate from the pancreas. CCK helps us make enzymes from the pancreas, but they're both helping us with bile salts. Secretin increasing bile secretion from the liver. The CCK is telling the gallbladder to contract and our sphincter to relax. So we get a bunch of bile in the duodenum, and we can then emulsify our fats. It's the thing that I'm focusing on, but this also helps us neutralize acid as well. There's some bicarbonate mixed up in there. Okay, so that is it. And next time we will finish up our understanding of the GI system by talking about peristalsis and segmentation, basically how we're physically